Hello, dear professor, and many thanks for accepting our invitation. It's a great honor for all of us. And welcome to all the audiences that are present in this lecture. I want to give you a brief introduction about the professor. Professor Murray is an associate professor in the Department of Psychiatry. He has completed a PhD in physics from Yale University with research in the field of computational neuroscience, followed by postdoctoral research at New York University in the Center for Neuroscience. In his lab, Dr. Murray concentrates on investigating the dynamics and function of neural circuits. Specifically, they develop modeling, theoretical, and data analytic approaches to systems neuroscience in close collaboration with experimentalists. Many thanks, dear professor, and we are ready for hearing your lecture.
It's really a pleasure to be able to speak with you. I'm very excited about this, and I'm really grateful for the invitation. And so, as in the introduction mentioned, so my background is as a physicist, and I work in neuroscience. My primary appointment is in psychiatry. And so my group is kind of at this intersection of kind of physics and computational approaches intersecting with basic neuroscience, with human neuroimaging, all the way to psychiatry. And that's basically the approach I'm going to tell you about today. It's how we can kind of take the perspective of using mathematical computational models of brain circuits and try to bridge across these levels all the way from the molecular to kind of systems level human neuroimaging. And so this is really our goal, right? And this is applicable for psychiatry, right? Where the challenge is really to bridge these very different levels of kind of the molecular level, which is where we have pharmacological therapeutics, all the way up to the level of symptoms and behavior where disorders are diagnosed, right? And we have multiple technologies to try to kind of bridge these up, right? We can look at the distributions of genes and receptors in the brain and see which circuits are involved by a disease process or by targeting with a drug. What I'm going to be talking about how we can use computational models of biophysical brain circuits to try to bridge from the microcircuit level up to systems, how we can test these at the systems level effect with neuroimaging and with pharmacological neuroimaging, and connect that to disease-related biomarkers and individual variation in symptoms. And that part I won't talk about today, but this is kind of the vision of how we can use different types of data and how we can use computational approaches to try to bridge across levels of analysis. Okay, and so the perspective that I want to take here is kind of building on this idea of looking at the human brain as this kind of large-scale network, right? And so we're going to consider kind of different nodes as different brain regions, say different cortical areas, and then there's some notion of an edge connecting them, right? Which is some kind of connection. And depending on the modality, this could be an anatomical connection of actually white matter connectivity. This could be some kind of functional measure like functional connectivity, the correlation of some non-invasive neuroimaging measure like a bold signal, et cetera, right? And the traditional picture is focusing on the properties of the graph itself, right? And the nodes themselves are kind of generic in some sense, right? And kind of all the information is in the kind of topological properties of the graph edges connecting them. And what I want to focus on, and I think this is very relevant for kind of connecting to cognitive function, connecting to disease, psychiatry, pharmacology, et cetera, is really kind of looking at the heterogeneity of the node properties within these large-scale networks, right? In other words, what kind of makes each local brain area unique and different from other areas, and how does that impact what we see at the large-scale network level, right? How does the local impact the global in that sense, right? And so the idea that we're going to take here, right? So it's this idea of kind of functional specialization of cortical regions. And so there's this idea of kind of the canonical cortical microcircuit, right? That at the microscopic level, kind of if you look under a slice, cortex looks like cortex. But we know that there are aspects of variation, right? There are layer differences, laminar differences in terms of the cytoarchitecture, cell type differences, receptor differences, et cetera. And we want to try to understand what the principles for that are and how that relates to, again, this kind of large scale whole brain systems level picture. And so if we look, for example, at the non-human primate brain, so this is a monkey brain, different colors are different cortical parcels. And one of the principles that we have for how these different regions pertain to each other is this idea of a cortical hierarchy, right? So this is kind of a classic figure from Feldman and Van Essen looking at the anatomical connections between areas. And they were able to kind of structure these areas from kind of low-level sensory processing up to association level as a hierarchy, right? And so our question is going to be, what are the aspects of functional specialization that we see across the hierarchy, and how does that relate to kind of the local properties of these regions, right? And so we know that there are a lot of functional differences, right? So, we know that receptive field sizes change as you go from early visual cortex V1 to V2 to V4, kind of up the ventral stream, you get cells which care about larger regions of space. That's an aspect of functional specialization across the cortical hierarchy. And there are also aspects that we see temporally. So if we compare a kind of a lower sensory area like MT, we can see neural activity which tracks stimuli in a very fast dynamic way. And if we go to a more association area like LIP, we can get very slow ramping dynamics across hundreds of milliseconds, right? And so one hypothesis is that there may be something different in the local circuitry of MT versus LIP of different cortical regions which support fast versus slow temporal dynamics, for example. And we also see this, for example, in looking at working memory. And my lab also works on computational models of working memory. And the key thing there is that you need to hold something in mind after a stimulus has been removed. And a sensory area like MT doesn't do that. It basically, represents the stimulus while it's presented and then falls back to baseline firing activity, whereas prefrontal cortex can hold the stimulus identity across the delay in this kind of selective persistent activity. And our computational models of this, do so by having the local circuitry be different. That prefrontal cortical circuit can have stronger recurrent connectivity, which then supports the persistence and reverberation of the signal in a way that a sensory area cannot do. And some early work during my postdoc actually tried to measure the timescales of fluctuations in spiking activity of neurons across different cortical regions in the hierarchy from sensory to association. And we saw this very consistent increase in what we call the intrinsic timescale as you went up the hierarchy. And so again, our hypothesis is this may reflect local differences in the circuitry, which can then support functional specialization for sensory processing versus association. Okay, so this is kind of background. So now, the kind of challenge today is, these are kind of principles that we observed in animals. And now the question is, how can we try to translate some of these up to the level of kind of, whole brain systems level human neuroimaging? And so I'm going to kind of tell 4 kind of brief vignettes, right? So one is, how do we even characterize the regional heterogeneity of human cortex? Then I'm going to talk about how we can build large-scale models of, in this case, resting state bold activity of the human brain. We're going to see whether that is able to capture individual differences, individual variation in brain dynamics. And then we're going to apply that to look at the effect of pharmacology. How can we actually bridge from molecular to systems by simulating the local effect of a drug and how that impacts brain-wide dynamics? Okay, so let's get started on the first part. So the first part is, how do we even think about cortical hierarchy in the human, right? Because what I showed you in Feldman and Van Essen, that was based on invasive track tracing in the monkey. And so what we looked to here was kind of a proxy measure that we can get from human just structural MRI, which may be able to track hierarchical variation. So here we looked at this, what's called T1, T2 map, which is just a structural contrast in MRI. It's thought to reflect the intracortical myelination level, how much myelin is in gray matter. And that's seen to be high in basically primary sensory areas in humans and monkeys, and then go to low values as you go up the hierarchy, right? So it's kind of an inverse proxy measure of hierarchy. The T1, T2 is high in sensory, low in association. And we can just kind of characterize that very briefly and see that indeed it has high levels in kind of primary sensory motor areas and in sensory functional networks. And then as you go into more association areas, this level drops. And so now we can kind of bring this into the human, and where we don't have an anatomical measure of hierarchy in the direct kind of way as we had from Feldman and Van Essen, we can use this as kind of an inverse proxy measure. Okay, so that's how we are going to characterize hierarchy, but then how do we characterize local circuit variation along the hierarchy? And for that, we used gene expression mapping. This is a public data set from the Allen Human Brain Atlas, which has gene expression in 6 post-mortem subjects. So they were able to sample bits of brain tissue post-mortem and run gene expression levels on that. And we're able to basically run that through our neuroimaging pipelines in order to get kind of whole cortex maps for different genes of interest, for thousands of genes that are in the data set. So we were able to kind of process that, and now we can compare that to our imaging measures, right? And so, we can look at different aspects of microcircuitry that we think are interesting and important. So we can look at, for example, at different synaptic receptors. And for a lot of them, we find very strong variation across this T1, T2 map. So this, for example, is the gene GRIN2B. It codes for the NR2B subunit of the NMDA receptor, which is thought to be important for working memory, for example. And we see the strong negative gradient with T1, T2, which means it has higher levels in association areas compared to sensory. And we see this in excitatory receptors, NMDA, as well as inhibitory receptors, GABA. And we also see strong hierarchical gradients in the expression levels of different inhibitory neuron markers, in particular for parvalbumin interneurons and calbendin and different cell types, which have different connectivity properties. And so this is just to show that, across this hierarchical measure, we see a lot of variation of the microcircuitry as can be kind of measured in this bulk gene expression. And we think that that may be important for the kind of functional properties of these microcircuits and the types of computations that they can produce. And so what if we throw just all the genes together, right? All kind of 10,000 genes. We can then do principal component analysis for dimensionality reduction and say, what is kind of the dominant spatial pattern of gene expression across the human cortex? And we get a pattern that, as you can see, looks very similar to this T1, T2 map, right? Very similar to this proxy measure of hierarchy. And on the left here, you can see that it's kind of quasi one-dimensional, that the first component picks up a lot of variance, right? So, in other words, you can think about it this way, that at the level of, gene expression, which is characterizing about what receptors, which cell types, what kind of microcircuit properties are there, the kind of, 0th order component would be, cortex is different than thalamus, is different than cerebellum. But within cortex, the kind of first order correction is this sensory to association hierarchical specialization of the microcircuitry. And given that that's such an important axis for functional specialization, for sensory processing versus cognitive processing, we think that the specialization of microcircuitry could be involved in that. And then again, this is going back to what I was saying about the large-scale network. These are properties of the individual nodes giving rise to functional specialization, not purely a result of the connectivity of what signals project from where to where, which clearly is important. But the local processing within each node can be specialized as well. Okay. All right, so how can we kind of now bring this to our models of large-scale brain activity in neuroimaging? So the idea that we're going to take, right, is that we have some idea of, again, the structural connectivity, right? In this network, how are different nodes, wired together? And we can call that, a structural connectivity matrix. In human neuroimaging, we can derive that from diffusion-weighted imaging, for example. And then the question is, how does that then give rise to the functional connectivity, right? The correlations in activity across the different brain areas. And that's the kind of mapping that we would like to build kind of mechanistic, physics-style models for, right? And so the idea is we're going to actually simulate some simple properties of, a local cortical circuit of excitatory and inhibitory neurons interacting. We're going to have some kind of physiologically interpretable parameters for synaptic strengths, et cetera. We're then going to, take that local circuit model of each cortical area, embed that in a large-scale network, which is then going to be constrained by the structural connectivity matrix, dictating how strongly areas are coupled. And then we'll simulate neural activity. That will then give us kind of a model-generated functional connectivity, which then we can compare to empirical patterns and actually do fitting of the synaptic model parameters to maximize that fit. That's the logic of the kind of, relatively simple style of modeling that we're going to be taking. Okay, and so in this study, what we did is, again, we took that approach. We're simulating local activity, simulating a bold signal, getting a model functional connectivity matrix. And now we're going to compare these two hypotheses, right? One is that we can treat every local brain area as the same, we call this a homogeneous model. So, there's the kind of canonical cortical microcircuit. We can use the same parameters for every cortical area. Or the alternative that we're going to test is what was informed by the gene expression mapping, that actually we have this hierarchical axis of specialization. And so what that means is the parameters in our model, like the strength of E to E recurrent excitation, cannot be fixed across areas, but could actually scale as we go from sensory association areas following this kind of inverse T1, T2 myelin map. Okay. Okay, so this is what we did, and then we can fit the model to the empirical functional connectivity. And so here on the left, you have the empirical structural connectivity. That's the scaffold of interactions of the different dynamical nodes in the network. We have the empirical functional connectivity as our target, and then we can see what kind of the best fit models are for that homogeneous model with local properties which are the same, and the heterogeneous model where local properties can follow this hierarchical gradient. And we can see how well they do. And what we found is that we can pretty substantially increase the fit to the empirical functional connectivity if we allow this hierarchical heterogeneity of parameters, right? So the goodness of fit, we do better than comparing functional connectivity to the structural matrix alone if we simulate the dynamics as we go to homogeneous, but we do even better if then we simulate these local variation following the hierarchy, right? And so the model is able to do this after fitting by increasing the local recurrent strength as you go up the hierarchy, which kind of fit with our prior hypothesis. And so you can ask the question, maybe it's a more complex model, it has more parameters. How do we know that it's really specific to this map? And so this is kind of an important methodological, but also conceptual control, right? And so what we did was figure out a method to generate alternative kind of random heterogeneity maps, which still preserves something of the spatial autocorrelation of our original T1, T2 map, but now it didn't give us a different axis, right? And so if just any axis of variation improves the fit, it may not be special to this hierarchical axis. And so then we can see here on the top right is the distribution of all these surrogates in gray. And what you can see here is that actually our heterogeneous model fit does in the kind of extreme few percent of that distribution. In other words, it does better than kind of a random topography. And in the bottom right, what we found is the degree to which other topographies improve the fit does so in a way that's predicted by how similar they are to our original map, right? So in other words, we got, maybe we got lucky, or maybe it was a good guess that we considered cortical hierarchy as a kind of preferred axis of heterogeneity. But we could have arrived there even through kind of random connections. Okay, I'm going to switch to the other slides to continue the talk. But actually, if there are questions, I'm happy to take a question now while we transition over. Otherwise, I'm trying to keep us within the hour on time. Okay, so I think we're doing okay there. All right, so now we have kind of a model framework, and we showed that we can improve the fit over this kind of homogeneous model, right? So now the question is, does this help us to, sorry, one second. Okay, so now we can ask the question, can we use this to actually model individual differences? So we're going to model individual variation now in this functional connectivity measure and see to what degree that improves our fit. So, okay, so the approach we're going to take, right, we're going to use that same modeling framework, except now instead of fitting an entire group of subjects, we're going to fit at the individual subject level, right? So in other words, the parameters are going to be optimized to fit an individual's fMRI resting state bold functional connectivity matrix. And then we'll do that for each subject. And this is using the publicly available Human Connectome Project HCP dataset, where we have hundreds of unrelated subjects. Okay, so our fit measure, just kind of a technical thing, this is what we're called coefficient of determination. It's just our kind of goodness of fit measure, which we can get into. And then what you see here on the left side are two individual subjects, and then the group average. And then we can show what happens as we fit the model to those individual subjects, right? And so you can kind of see visually that we're able to, fit something about the individual differences using this, again, kind of constrained biophysical circuit model, right? And on the right here, you can see that here we're comparing the goodness of fit for the heterogeneous model, which again has this hierarchical gradient, to the homogeneous model where we don't. And you can see that, basically each dot here is a subject, and basically every individual is going to be better fit if we allow that hierarchical access. And so, one hypothesis is that actually the sensory dissociation hierarchical gradient is actually an important aspect of individual variation that we see. Okay. So we can do some other things to see how much it improves things to fit at the individual level versus the group level. We see a strong improvement in fit. We get a better fit for the individual level compared to the group. And we can see that we get a better fit when we fit to that own individual versus a different subject, right? So it's just kind of a sanity check that we are able to get important differences. All right, but the kind of bigger scientific question is, what are the kind of patterns or modes of individual differences, and how can we model those, right? And so to do that, we here did principal component analysis to say what are the patterns of variation, but now it's patterns of variation on the functional connectivity elements, right? And so here when we do PCA, we kind of get certain modes, and we can basically look at those and try to interpret what they mean, right? And so our empirical PC1 has this on the bottom left here, has this kind of global increase, right? In other words, the kind of dominant way in which individuals differ in their functional connectivity matrix is in this kind of global shift, right? Everything's increasing or decreasing together, which looks like global signal, right? So we can kind of interpret the first principal component as related to global signal. The second component actually ends up kind of looking like hierarchy. It ends up looking like this kind of T1, T2 map. And so we can interpret the second mode as reflecting hierarchical differences, sensory dissociation differences. And then the third principal component, the third most important one, actually kind of looks in this kind of block diagonal structure, looks kind of like functional networks, right? And therefore we can kind of get this hypothesis, and this can then inform our modeling, that principal component 1, PC1, is about kind of global coupling. Principal component 2, the second way in which subjects differ, is about sensory dissociation hierarchy. And then principal component 3 is about network structure, right? And so the question is, what do we need for our models to be able to capture this as well? Okay, so the approach that we're going to take is kind of a state space geometry view, right? So in other words, what we're going to look at is as we perturb, as we vary the parameters of our model, right? And our circuit model produces a bold functional connectivity. We can say kind of how much sensitivity is there under my parameters. So another way to say this is, the experimental mode of variation that we see, right? The experimental principal component, 1, 2, 3, can that be captured by changing the parameters of my model, right? Does my model have sufficient expressivity to capture that mode of individual differences in the empirical data? Does that make sense? And so we can quantify that by actually kind of deriving what we call a sensitivity subspace of how much of the functional connectivity can be captured by changing the model parameters, and then seeing whether the empirical principal component kind of lies within that subspace, how much of it lies within that subspace. And so when we do that, again, here we're comparing the homogeneous and heterogeneous models, right? Whereas the heterogeneous, again, has that hierarchical gradient. What we see is that the first principal component, the one that looks like global signal, can actually be very well captured by both, right? So in other words, just like the global coupling of the strength of connections across the nodes in the network is enough to capture the first principal component of variation, which is good for the homogeneous model. But when we get to the second principal component, that's actually the one that now we're much better able to capture by the heterogeneous model compared to the homogeneous model. And therefore, this gives us kind of a rational way to think about how to expand the models, because now we can say that, that including the hierarchical variation is important for being able to capture the second principal component of individual differences in functional connectivity. And so that gives us a kind of strong rationale for why this is a useful thing to have. Okay, so can we take it one step further, right? Can we use that same logic to tell us what would be a useful future expansion of the model? And so for that, now we can look at principal component 3, right? And so we can, and there again, we saw that the pattern kind of looks like functional networks. And so the idea is that then we can say, okay, well, if we introduce now another parameter into our large scale model, which controls, here we looked at the structured input, so kind of common input to different nodes. If that follows this kind of network structure, then that then allowed the model to capture this mode of variation. And that's what we see here on the bottom plot, that our heterogeneous model, which didn't have this kind of network structured noise, again, it could capture PC2 better than the homogeneous model, but it didn't do a good job at improving the fit to PC3. But now if we add this kind of network structured noise, now we can improve our fit to PC3. So this kind of gives us a way to evaluate, how complex should these models be, right? And if we want to, we could throw in every level of biological detail that we know, or we can try to keep them more parsimonious and interpretable. And, one way to interpret how much we should be adding in, how many parameters should we have in the model? Is this a useful parameter? Is this not a useful parameter? Looking at something like, does this model give us the expressivity to capture individual variation, individual differences, is kind of a good benchmark to interpret the parameterization of our models. Okay, so the last part of the talk is now, trying to bridge from kind of molecular to systems through pharmacology. And so the logic there, right, what we'd like to do is be able to take this large-scale circuit model where we have some level of biological detail at the individual kind of node microcircuit levels, where we actually can, simulate the effect of a drug at a particular synapse or a specific cell type, where we can use the large-scale gene expression mapping that we derive from the Allen Human Brain Atlas to talk about which brain regions, which cortical regions are preferentially targeted by the drug due to their density of the different receptors. and then be able to put that all together in the model and then make differential predictions for some neuroimaging measure like fMRI or EEG, MEG, based on, you know, to dissociate the effect of different receptors, which may be targeted pharmacologically. And the kind of big picture view of this, right, is, you know, if we think about like a psychiatric disorder, you know, that's going to have a specific neural kind of biomarker pattern to it. And if we can understand the mapping of what the effect of a drug is, a therapeutic, then maybe we can kind of match these up, right? Maybe we can use these kind of brain-wide patterns to kind of rationally design which therapeutics, which, you know, drugs, which molecules being targeted could be useful in order to induce a specific brain-wide pattern of, say, change in functional connectivity, which might be then, you could say whether that is aligned or not aligned with the type of biomarker that we see in the same measure for a given psychiatric or neurological disease. Okay, so that's kind of the big picture, the goal. So we started as kind of a test bed with an existing data set that some colleagues had collected, and this is using LSD. So this is this is actually collected in Switzerland. You can't really do these experiments in the United States with LSD, but in Switzerland you can. And so they took healthy subjects and they gave them this, you know, serotonergic hallucinogen, LSD, and it induces a specific kind of brain-wide pattern in the functional connectivity. And here we're using a measure that we call global brain connectivity, or GBC, which is just kind of the average functional connectivity for a given node, for a given cortical region, you basically collapse across all the other areas, and that gives us a single number per area. And so therefore we can say, what is the change in GBC? What is the change in mean functional connectivity under the drug? And so here we get this kind of structured brain-wide topography. In particular, we see these large increases in visual cortex. Maybe that's related to kind of visual hallucinations that we associate with some of these hallucinogens. And then what was actually quite unique about this data set is that they were able to completely block the neural and behavioral effects of the drug by pre-administering with catanserin, which is a more selective antagonist of the 5-HT2A receptor, right? And so LSD is, thought to be, potentially targeting multiple receptors in the human brain. It's a little unclear what the key receptors are for this neural effect that we see. But this gave us then a very strong hypothesis, right? That it's actually, this pattern we see is due to a single receptor, the 5-HT2A receptor. And the spatial topography that we see across cortex then gives us a clear hypothesis, right? Which is that this neuroimaging map should align with the gene expression, going back to the earlier part of the talk, for that particular gene of HTR2A that codes for that specific receptor that we know is involved. Okay, so what do we do? So we took this resting state fMRI map from healthy subjects administered LSD in Switzerland. We compared it to these postmortem gene expression maps from the Allen Institute. And then we looked at the correlation, right? And so for different kind of candidate genes of dopamine or serotonin, we found that in line with our hypothesis, HTR2A had the strongest positive correlation. And indeed, if we look at the distribution of kind of all the receptors in the Allen Human Brain Atlas, we see that the HTR2A is in the top few percent. Okay, so that's just kind of jumping from the gene to the neuroimaging, pharmacological neuroimaging. Can we model this, right? And so we're going to, again, use the same kind of circuit model approach as I showed you. We're going to embed these local microcircuits in a large-scale network, constrained by structural connectivity, simulating synaptic and bold activity through a hemodynamic model. And now we're going to kind of simulate the effect of the drug, right? And within, again, a relatively simple model, right? And so based on the physiological literature, we think that the HTR2A receptor is basically modulating the gain, the input to output relationship of a local region, right? And so we get an increase in gain, and we're going to have the strength of that gain modulation for each cortical area be modulated by the gene expression map, right? In other words, a cortical region which has a larger value of the gene expression, we're going to give it a stronger gain modulation. And then within the local circuit, if we have E versus I, we're going to allow that to be fit as free parameters, whether E cells or I cells are preferentially targeted. And so that's then going to give us a simulated bold delta GBC out of the model, and then we can compare that to what we see empirically. Okay, and so when we do that, and we can vary these two free parameters, which is the excitatory gain increase and the inhibitory gain increase, again, following that topography, what you see is we get this kind of ridge of best fit. So there's some degeneracy, but you can see that it's shifted to the right of the diagonal. And if we collapse across all of this, what we see is that the best fit occurs, it's basically determined by kind of EI ratio, right? The ratio of the change in excitation over change in inhibition. And we see that's shifted to the right, which means that this is best fit when the effect of the drug is on net kind of increasing excitability, increase EI ratio through a gain increase on the pyramidal neurons in the model, right? And that's, I think, consistent with physiological hypotheses. So again, we can then see how good of a fit we get, and we get a pretty good correlation. You see here a correlation of 0.73, and on the right here, what I think is most interesting is that it shows that we can do better than the gene expression map alone, right? That by simulating the signal that we're actually measuring empirically, we can do better than just the map alone. And again, we're able to kind of fit various aspects in terms of sensory versus association differences, which is quite cool. Okay, so I'm going to wrap it up there so we can have some time for questions. And so, in general, our kind of effort is to try to bridge these levels through kind of mechanistic models to actually be able to use knowledge of, local neurophysiology, and the molecular effects of pharmacology and bridge that all the way to human neuroimaging and ultimately to inform psychiatry and therapeutic development. And so we think that this is kind of a useful platform that we can iteratively expand and refine and add in more biological detail as is important for the questions at hand. We think this is going to be useful for fitting at the individual level. And that's important because, when you're looking at disease, at psychiatric disorder, we see a lot of heterogeneity, right? And this is one of the big challenges in psychiatry, is how can we kind of parse the heterogeneity, right? So within a disorder like schizophrenia or depression, you can have a lot of symptom variation, even though they have the same diagnosis. And you can get a lot of variation in terms of whether subjects respond to a given, therapeutic course or respond to a given, pharmacological treatment. And we don't really have a good way to understand that rationally, to identify subtypes or dimensions or have some way to predict who's going to respond well or who's not. And so, being able to do these types of analyses and modeling at the individual subject level is, I think, very important. And again, ultimately, we're hoping to use this as a kind of test bed for looking at disease and looking at pharmacology. Okay, so I'd like to acknowledge my main collaborators on these projects, our funding sources, and I'd like to thank you for your attention. And now we can take some questions.
Thank you very much, Professor Lida. Yeah, please go on.
Yeah, thank you, dear professor, for this amazing and insightful lecture. I want to invite the audiences and attendees of this lecture to ask the questions. You're ready for hear the questions. Everyone who wants to ask questions or raise the hands, you can type your question through the chat box also.
Yeah, please.
Yeah. So during the preparation of the audiences. Dear Professor, I wanted to ask one question. As you explained about the large-scale brain networks and the dynamics of the neuronal activation throughout the brain, and also the neurobiological underpinning and receptor function for creating these dynamics. My question is that, how about how we consider a role and a contribution for other types of neuronal transmission and neuronal communication, like apoptic transmission, or specifically apoptic modulation of chemical synapses? As I know, recent literature shows that these kinds of transmission also can affect the hierarchy through creating a spatiotemporal complexity. Thank you very much.
Yeah, so it's a very good question, and you know, really there are, you know, there are like more things which are missing in the model than are included in the model, right? And I think that, you know, this is a situation where, you know, this is also why, you know, we need experiments and modeling in animal models ultimately as well, right? Because, you know, not everything we're going to necessarily be able to, you know, constrain purely from the level of human neuroimaging. But again, may be really important, especially, you know, when you're, depending on what measures you're looking at. So in the human, I think this is also a place where combining modalities is going to be useful, right? So in one thing that we did in this earlier paper is we took this hierarchical variation, and that also made predictions for kind of faster time scale power spectra beyond what we could see in bold. And so there we actually looked at a public Human Connectome Project data set from MEG. And what I think is really interesting, and what we're trying to do now, you know, people can do, you know, you could do EEG and fMRI in the same subjects, and you know, in some facilities are able to do that simultaneously. That you can actually do EEG cap-on in the fMRI scanner, and that would then be a way to, I think, really constrain the models with kind of frequency content as well as the spatial content. And I think that's going to be an important way forward in terms of, as you said, kind of adding in more of the biological complexity, which we know is present in these microcircuits. But, you know, how do we constrain that, right? And, you know, if all we have is, you know, a bold functional connectivity matrix, maybe the coarseness of the model that I showed is sufficient for that. But now if we introduce frequency content, now if we introduce maybe in non-stationary dynamics or the effect of different types of perturbations, that will be useful to help us, you know, build more complex nodes that include these other properties. It's a great question.
So thank you very much, dear Professor. I see a Muslim, you have raised your hand. Please ask your question. Turn your mic on and ask your question. We can hear you. We are ready for your question. So, Dr. Batuli, could you please?
Yes, yes. In the meanwhile, I'm having a lot of questions. First of all, I should thank for a very interesting and also complicated talk. I was trying to cope with the talk, so not to lose anything. Thank you very much, Professor, for the talk. And just one suggestion: if you have problem testing something like LSD and you have to go to Europe, just come to Iran and you can try many different things simultaneously. No problem here. Yeah. Just one question. You are speaking about the global signal. Can I ask, when you're using the principal component analysis, what does global signal represent? Which brain network? I mean, does it represent any particular brain network? Is it default mode salience or not, or anything like that?
Yeah, so it's a good question. And, you know, global signal is can be tricky to interpret because, you know, there's a lot of kind of debates in neuroimaging about to what degree that may be confounded, and, you know, to a degree that has a neural origin versus, you know, respiratory or something like that. And people try to, you know, obviously remove confounds as well as they can. So, I mean, you know, the most notable thing about it is that it is kind of global, right? That it is all nodes kind of moving in the same direction together. But you're right that within that, there actually is a topography to the global signal, which is, I think, higher in sensory areas than association regions. And that's something that in our 2019 paper, Demirtas et al., we actually looked at that in terms of, because it's reflected in like kind of the global, mean GBC value, that if an area is stronger in the global mode, it will have a stronger mean functional connectivity overall. So, yes, maybe that's the answer, is that, you know, mostly it's kind of global. It really is all kind of regions kind of statistically moving together. But there is some topographic variation, and there are a lot of kind of debates in the field about how that may be neural or non-neural. But I think this is, you know, an important thing that the modeling needs to be able to kind of take into account, right? But at least it demonstrates empirically, again, that this is actually a major aspect. The dominant aspect of individual differences is just mean functional connectivity, which is maybe not surprising. And that, you know, the models demonstrate that there can be neural contributions to that, and show kind of how that can arise. Just, you know, in the models, you can really just get it through the average. You can just scale up the strength of coupling and get something like that.
Thank you very much.
Thank you, dear professor.
Can you hear me?
Thank you.
Yes.
Yes, Muslim, we can hear you.
My
Ask your
name is
question.
Muslim.
Yes.
Thank you for your representation. I've read your book on computational psychiatry, and I learned a lot from it. Thank you for writing that book. My question is about chapter 4 of that book, which was also discussed here, where you introduce some analysis such as dynamic mean field or chromotal.
Yes.
I have looked at famous toolboxes such as SPM, and there is no option for this analysis. Is there any toolboxes to implement this analysis, or do we have to code manually? Thank you.
Yeah, so actually, I mean, I edited the book. I didn't write that particular chapter, although postdoc Murat, I think, wrote that one. Yeah, so let's see. I guess there are two answers to that. Number one is the kind of standard in the field. Most people actually would kind of code up the simulation themselves, which is, I think, not actually too challenging. If you want to look at something, I would look at there's something called the virtual brain, TVB, and this is a large, well-funded initiative out of Europe as kind of a framework for doing these large-scale models in relation to data. My personal perspective from kind of a research side is that it can be a little bloated. It actually has like a lot of features, which is often kind of more than one needs for the types of research questions, but it can be very useful for kind of visualization. And I believe that they do have multiple types of different node models. So I think that they do have dynamic mean field. They do have, I assume they have Kuramoto. And so that may be a good thing to check out. It's called the virtual brain, TVB. So I would check that out.
Thank you very much. Thank you, sir.
I've never used it myself, but it's like a big part of the Human Brain Project in Europe.
Thank you, Prof.
Thank you, Muslim. So I can see another question. Dr. Batuli, do you have any other question?
Yeah, of course, I can ask for 1 hour my questions. One other question is that when you are modeling the brain, do you consider different hemodynamic response functions for the different parts of the brain, or just one HRF?
Yeah, it's a good question. That came up in review of our paper. So here we are just using a single model. We're using the so-called balloon Windkessel model, which has been used by people like Carl Friston and put forward. And so, we didn't really innovate at that level. We just kind of took what seemed to be the kind of leading model for that hemodynamics. This is from Carl Friston and Klaus Stefan, et cetera. So we did not include variation, which actually I think are really, we looked into that literature. I think it's a very hard thing to put in for the following reason, right? So there's a bit, to my mind, there's a bit of conflation in this idea of HRF differences across areas, right? Because some of that could be actual due to regional differences in the vascular coupling But also what people call individual HRF differences also due to the time course of the neural activity, right? That if you have a longer activation in hippocampus versus V1, that turns into a longer HRF, which in hippocampus, right? Which is useful if you're actually doing, event-based PLM modeling. If you're actually trying to build a mechanistic model of, say, resting state activity, the longer activation in hippocampus is because the neural activity is active longer in hippocampus, right? Not that the hemodynamics are necessarily. And so we thought it was important to actually kind of keep the hemodynamics fixed and see what can be explained by the regional variation in the neural activity itself. And I think in terms of actually constraining differences in the hemodynamic coupling, I think we're probably going to need people to do experiments in animals, right? Where you could do like an optogenetic, in other words, you really have to control the neural activity to be the same in different areas to then be able to resolve the differences in hemodynamics, because the kind of HRF differences that we see kind of combine both of those two things, right? Differences in activity time course and the differences in hemodynamics. And so I don't think that in the human that's really dissociable.
Yeah, but I think there are projects which are trying to understand how the HRF is different in different brain areas. For example, by using the CO2 gas, by increasing the amount of carbon dioxide in the blood, and I think there are projects, but they're in the very beginning.
Okay, yeah, maybe I need to learn more about that. But I mean, yeah, my point is just that you have to be able to separate out, because I think there are going to be regional differences in the neural activity, and you have to control for that if you want to talk about regional differences in how that neural activity couples to hemodynamics. So that's
exactly.
Yeah, I agree. I think it's a very open question.
Yeah. So if Lida, if there are other questions coming, or if you have any other question, just please ask me. I don't just want to make the professor too much tired.
Yes. I have another question, but I do research. Yes, thank you very much. I do research in neuropsychopharmacology, specifically neuroimaging of neuropharmacologic effects. But sometimes there is significant gaps between the experimental area and the clinical practice of the neuropsychiatry and psychopharmacology. Sometimes I feel it's imaginary. Someday it could be incorporated into clinical practice. Or how do you see the future of these fields?
Yeah, I think it's still very much kind of. I agree with you. It's still kind of promise more so than application at this point. But I think in large part it's because we, kind of don't know enough about the effects of drugs on the brain, right? And this is true like in drug development, right? I mean, right now, I actually, talk with people in industry, and, they have a disorder that they're interested in. The kind of typical situation is the following, right?
Yeah.
And this is actually, relates to the status of like drug development for psychiatry, which is kind of, it's people kind of describe it as in crisis right now because the rate of failure is so high of clinical trials in psychiatry. And and one big question which we're very interested in pursuing is, can brain imaging help with this, right? Because the status quo is like we have a bunch of drugs that we know get into the brain.
Yeah.
We know that we've done safety. We know that they get into the brain, but we don't know who to give them to.
Yeah, exactly.
And so, and so much of what we're doing is just kind of in going blind, or the trials are failing because of patient heterogeneity, right? That maybe post hoc we can see some people improved, some didn't, but we don't have a good way to kind of parse that beforehand. And so, and brain data is not being used at any of those steps before you get into phase 2 trial failure, right? And so the question, and so if we had the brain maps of what brain circuits are being engaged by the drug, if we had large clinical cohort data sets where we can say, what is the heterogeneity within that, and do different dimensions of symptom variation predict different circuit patterns of dysfunction?
Yeah.
Do we have the opportunity of aligning those up and then making some hypotheses which can improve our success rate of saying, you know, the circuits that are engaged by the drug, if we, you know, kind of refine our clinical sample to have the symptoms that produce that same pattern, that an aligned pattern of circuit variation or disruption instead of a different one. And that's never really been tested. And so maybe it's an overly simplistic hypothesis, but it's, you know, it's still more evidence-based than I think a lot of the status quo is.
Thank you very much, dear professor, and thank you for providing this situation, for asking this question and this consideration. Thank you very much.
Great. Thank you. Thank you. It's very nice to be able to speak with you.
Okay.
Bye.
Dr. Ashton, please, yes. And thanks.
Of the professor, and it was indeed an honor for us to have you today with us, Professor. Thank you very much. Sorry, students and friends. I know you have more questions, but we cannot make the professor too much tired. Thank you very much again.
Thank you.
Bye-bye.
Bye.
Have a nice day. Bye.
Yeah, bye.